we saw some big picture ideas for DNA. So now we're going to zoom in because it's a chemistry class and look more at the specific molecules. We'll be using the same color coding. So in this segment of DNA that we're looking at here, you'll see that there are some bases. And there's two different types of bases. There's purines. The purines have double rings. You don't need to recognize the details of them, but you should know that the purines have two rings connected. And then also pyrimidines have a single ring. So pyrimidines have a single ring. And you can see here's a double ring and a single and a double. So here's my bases in the middle of this DNA molecule. Don't forget, like we saw on the last slide, that we have hydrogen bonding happening in the middle here, holding these together. And if I look at the atoms, you know enough now to be able to notice that, oh yeah, that makes sense. So here's a nitrogen, and here's an H connected to a nitrogen. So we have a donor, NH, and an acceptor, N. That's what I need for hydrogen bonding, right? Or here's an oxygen, acceptor, and then NH2, which can be a donor. So I have a donor and an acceptor, and that forms a hydrogen bond. So my purple connections as I go through here are all hydrogen bonds, and that's what's holding my base pairs together. Remember, A's pair with T's and C's pair with G's. Cytosine, guanine, adenine, thymine. The ene, remember, is coming from the fact that these are all amine functional groups, and so we see that ene ending to them. So those are my bases in the middle. We also saw on the last slide the sugars, and this is DNA, so the D stands for deoxyribose. If you look at deoxyribose compared to ribose, you can see right here that one of the OHs is gone. So that's why it's a deoxy, or loss of an oxygen in the sugar. So as we look at these sugars, notice there's a little empty gap on each of them. So these are all my sugars, and they are all deoxy, so lost that oxygen at that position. So those are my sugars and my bases. Remember, a nucleotide was a phosphate and a sugar and a base. And then we said that there were some phosphodiesters and phosphoester bonds. So let's see if we can find those as well. So if you remember, an ester is a carbon that's connected to two oxygens, and then it's got other atoms surrounding it. So this was an ester. A phosphoester is going to be very similar, except it's got a phosphorus in place of this carbon right here. If it's connected in one direction, we call it an ester. If it's connected in two directions, we call it a diester. So let's see if we can find some examples. So here's a phosphorus that's connected to a carbon. So this is a carbon here. It's only connected in one direction to a carbon. So this is what I would call a phosphoester bond. Those are high energy bonds. They're also going to be forming the backbone of my DNA here. Here's a phosphorus, and it's connected up to a carbon and then down to a carbon. And so because it's connected in two directions to those sugars, this is a phosphodiester bond. So the diester connects to two rings, and just the ester connects down to the one ring. So here is a phosphorus. Is that di or regular ester? See how it's a di? Two rings, di. Here, two rings, di, two rings, di, two rings, di, and then just one ring. So these are all diesters, phosphodiesters, and then not a phosphodiester here. Again, two, 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 and then just a single phosphoester bond here. And we'll see that those phosphoester bonds actually show up. This is ATP down here. Adenosine triphosphate, and here's some phosphoester bonds through the middle of that molecule. So just to draw it with circles here, a phosphodiester will have a sugar ring, and then a phosphorus, and then a sugar ring. So it'll have sugars on both sides of it, and then a phosphoester will be a phosphorus just connected to one of those sugars. So that's what we're looking for in terms of labeling that. One other key thing I want to show you here are these numbers. So we have the 5' end and the 3' end, and you saw that on the last slide. I had written down this 5' to 3'. So DNA strands have directionality, and those numbers we've actually seen before in chapter 14. So if I look at the sugars, if you remember with our sugars, this oxygen, carbon, nitrogen is very similar to an acetal functional group, and we would call that carbon number 1. If I keep going, 1, 2, 3, 4, and outside of the ring to 5, where there's a free hydroxyl at the third carbon, that's my 3' end. So this 3 counting on the sugar is where that 3' end comes from. If I go up to the other end, it should be a 5' to 3', and we often will draw that as an arrow. But look at the 5. Let's go back to the sugar. So here's that functional group between the O and the N. That's carbon 1, 2, 3. Here you can see carbon 3 is tied up in a phosphoester bond, phosphodiester bond. It's not free like I saw here. So down here I had that free hydroxyl at carbon 3. Up here I'm counting around 1, 2, 3, 3's tied up, keep going, 4, and then the carbon outside of the ring is carbon 5. And 5 is tied just to a single phosphoester bond. And that is why it's called the 5' end, because that 5 carbon is essentially free or just connected to a phosphate. So at the 5' end, we'll always see a free phosphate, and at the 3' end of a DNA strand, we'll always see a free hydroxyl. And we always see that our strands are anti-parallel to each other in DNA, so we're going to go from the 5' end, phosphate to hydroxyl, and then the opposite on the other side, from the 5' up to the 3' end. And so those come from our sugar numbers that we saw before. Last connection to make here is looking at this ATP molecule. We can see nucleotides here, but we can also see nucleotides, and we're going to see these in chapter 18, where it's still a phosphate sugar base in a totally different setting. So can you see the base here? Here's my base. This is actually adenine, the same A that we see here. So adenine, and then a sugar, and then my phosphates. One little thing I want to point out to you, notice this OH is not missing. So this is actually the sugar ribose in ATP, not deoxyribose like I can see here in the DNA. So be able to pick out and label any of those structures. And then we'll see more nucleotides used for entirely different things than DNA in a future chapter.